My name is Pallavi A. from Maharaja Institute of Technology, working as an assistant professor in Department of Chemistry. As we know, under VTU syllabus for the computer science engineering stream, we have sensors and energy systems as the first module. So let us know something about sensors and its function. So what is a sensor? A sensor is a device which is capable of giving analytical information about a test sample. So this sensor interacts with a specific analyte, detects it and produces a signal proportional to its quantity. Then what is analyte? Analyte is the target species which is being detected and measured using the sensor. Let us know the basic components of this sensor. There are three main components of sensor. They are receptor, transducer, electrical signal and display. Electric signal and display. Let us know them one by one. So what is a receptor? Receptor is a recognition element which is capable of interacting with analyte specifically and selectively. Receptor is the one which produces a signal corresponding to the interaction in the form of potential, conductance, current, mass, heat, pH, color, etc. It may be any parameters. If the receptor is of biological origin, the device is said to be a biosensor. If the receptor is of chemical origin, it is said to be a chemical sensor. So this is about receptor. It is called as the recognition site, whereas transducer is used to convert the signals created by the interaction between receptor and the analyte into readable and measurable values, which are the physical quantities. So where receptor is the recognition site responsible for the recognition of the sense, transducer is used to convert signals created by the interaction between receptor and analyte into readable and measurable values in physical quantities. So what is electrical signal and display? The electric signal analyzes the signals given by the transducer, helps in the signal amplification and converts the signals from analog to digital form. So this is the basic concepts about sensor having receptor, transducer, electrical signal and display. In the interaction between the receptor and the analyte, it can be monitored by several methods. Different parameters are used in which each method has different own functions, like change in potential, change in conductivity, change in current, change in mass, heat, pH can be measured using different sensors. These are several types of sensors available depending on the different transducers which are responsible for the measurement of different parameters via different sensors. Let us know electrochemical sensor. So the type of sensor which we are going to know is electrochemical sensor. Electrochemical sensor is a type of sensor which converts the effect of electrochemical reaction between analyte and electrode surface into useful signals known as electrochemical sensor. So the interaction between analyte and the electrode surface is converted into readable or measurable values. In electrochemical sensors, the electrode is used as a transducer element. As we know, electrochemical sensor is used to measure electrochemical change which is classified into potentiometric sensor, amperometric sensor and conductometric sensor. Let us now know the working principle of electrochemical sensor. Electrochemical sensor consists of a transducer element covered by recognition element. The recognition element interacts with the target analyte and signal is generated. Electrochemical transducer transfers the chemical changes into electrical signals. The electrical signal is related to the concentration of analyte. Electrochemical sensor always requires a closed circuit. Current must flow within the circuit to make a measurement. Since we need a closed loop, there should be at least 2 electrodes which are necessary in this electrochemical sensor. These sensors are often called electrochemical sensors because of the presence of electrodes and the change in potential which could be observed. Under electrochemical sensor, the first type of sensor is potentiometric sensor. In this sensor, change in potential during the chemical interaction between receptor and analyte is measured using the combination of an indicator electrode and a reference electrode. So, in potentiometric sensor, there are 2 electrodes which are the indicator electrode and the reference electrode. Commonly, platinum is used as the indicator electrode and the standard calomel electrode is used as the reference electrode. Indicator electrode is used to measure the change in potential due to redox reactions occurring on the surface of the electrodes. Each glass electrode is an example of this type of electrodes in which the platinum electrode is used in the measurement of current. But in potentiometric sensor, measurement is taken at zero current. So there is no current applied in terms of potentiometric sensor. Under the types of electrochemical sensor, amperometric sensor is the second type of sensor, which is similar to potentiometric sensor consisting of electrolyte solution in which the electrodes are immersed and the 2 electrodes which must obviously be present. But amperometric sensor is operated under externally applied fixed voltage. So, there is potential which causes the analyte to react and the current to pass. At a fixed potential, the magnitude of current generated is directly proportional to the concentration of analyte. In amperometric sensor, the current passing through the cell is measured. So this occurs in the presence of current, whereas potentiometric sensor occurs in the absence of current, where we could say zero current or no current is used. Here comes the third type of electrochemical sensor, that is the conductometric sensor. Conductometric sensors are the type of chemical sensors that measure the change in electrolytical conductivity of the given test sample or the analyte, which is occurring between the 2 electrodes dipped in the electrolyte. So, the principle of conductometric sensor says that in a conductometric sensor, it is composed of 2 electrodes which are coated with a highly sensitive material which responds to the change in the specific conductance of the analyte. So, when an analyte meets the electrode coating, it binds to the surface, causing a change in electrical property of the sensor. The change in conductivity is measured and correlated with the concentration of analyte in the sample. The final output is a quantitative measure of the concentration of analyte, which can be finally obtained in the form of digital. The conductivity is a result of dissociation of an electrolyte into ions. The conductivity, as we know, it is the result of dissociation of an electrolyte into its respective ions, cations and anions, for example. The migration of these ions is induced by electric field. When a potential difference is applied to the electrode, there is an electric field with the electrolyte. So, positively charged ions move towards the cathode and negatively charged ions move towards the anode. The opposite attracts. Thus, the current in the electrolyte is caused by the ion movement towards the electrodes, where the ions are neutralized and isolated as neutral atoms. This chemical change is recognized by the working electrode, and then the transducer converts this chemical change into electrochemical or electrical signals. So, conductometric sensor is a type of sensor in which the chemical or the electrochemical change can be absorbed, detected and measured. This is how the working of conductometric sensor occurs. Let us now know the applications of conductometric sensor. Conductometric sensors are used in the monitoring of water quality. They are used in the detection of gases, vapors and various other components. Conductometric sensors are also used in measurement of ionic strengths. They are used in detecting pH levels. They are used in food and beverage industries and various other sectors. Also used in the analysis of biochemical compounds. So the next type of sensor which we are going to study is optical sensor. Optical sensors are the electronic components which are designed to detect and convert incident light into electrical signals. The incident radiation from the sun or from the source is converted into electrical signals, which is finally then displayed. Calorimetric sensors are the example of optical sensors. So, under the optical sensors, we are going to study its principle, working and applications. When a sample of solution is interacted with the light of suitable wavelength, the certain quantity of light is absorbed by the analyte solution, which is then absorbed by the sensor. The transducer converts this intensity into electrochemical signals. The electrochemical signals is the change in potential. It is the parameter. So, the change in intensity at certain wavelength within the visible region can be determined using specific instrumentation. So, in an optical sensor, the sample solution is interacted with the light of suitable wavelength, under which the known quantity of light is absorbed by the analyte, and it is then being detected by the transducer of the sensor, which is further converted into change in potential or electrochemical signals. This is the principle of optical sensor. Let us now know the working of optical sensor. When a monochromatic light is made to pass through the analyte solution, certain quantity of light is being absorbed, and it is the function of a concentration of analyte. The change in the intensity of light is detected by the photodetector. The light source generates an intense radiation signal which is needed to detect the optical properties and the molecular recognition element which is in the sensor. The amount of absorbance can be governed by the Beer's-Lambert's law. So this is about optical sensor, whereas optical sensor has various enormous applications in different sectors and fields in medical diagnosis, in biomolecules detection, in cholesterol detection, in hemoglobin detection, and whereas in blood and urine levels, checking the amount of sugar, presence of different various amino acids, detections. So optical sensor plays a major role. Optical sensor is also used in security and system, in food safety and also food testings, and in detection of various gases like CO2, O2, etc. The next type of sensor which we are going to study is thermometric sensor. Thermometric sensor is a type of sensor which is depend on the measurement of thermal changes which occur during the interaction between analyte and receptor. As we know, analyte and receptor, they are the major components of your sensor. So thermometric sensor is a type of sensor in which we measure the thermal changes which is occurring between the analyte as well as the receptor. Sense changes which are converted into readable or measurable form is called the potential. So in thermal sensors, the change in potential can be in the form of change in temperature. Thermometric process is possible only in those processes which generate sufficient heat, and the change can be measured in the form of temperature. So, chemical or biological species which undergo different various catalytic reactions, chemical reactions, enzyme catalyzed reactions, all such reactions can be determined using thermometric sensor. This is about thermometric sensor. Let us know the working principle of thermometric sensor. The main component of thermometric sensor is a small tubular catalyzed reactor. The main component of this is a small tubular catalyzed reactor which is fitted with a temperature transducer. The walls of this reacted metal or the reactor is coated with the catalyst or enzyme. As we all know, enzyme or catalyst is the one which we are using for to increase or decrease the rate of reaction, which can alter the rate of reactions. So the walls of these reactors is coated with either an enzyme or a catalyst. The analyte is fed into the reactor. That means the analyte is put or fed into the reactor, and the catalyst interacts with the analyte which liberates heat. So the heat is liberated. The liberated heat is then quantified in the means of change in potential or voltage by the transducer, which is then amplified and further displayed. Thermometric sensors plays various applications in determination of metabolites, bioprocesses, and various environmental controls. It is also used as combustible gases, and it is used in hazardous locations like various power plants, etc. Until now, we have come an overview of sensors, various type of sensors, their application in various fields. Now, it is this time where we study the applications of sensors in pharmaceuticals. The use of sensors in pharmaceuticals, for example, here we take diclofenac. We know it is an antipyretic drug. As we all know, we'll study the detection of diclofenac using sensors. Let us now know the use of electrochemical sensor in the detection of diclofenac. So, in the detection of pharmaceuticals, we basically know there are many organic compounds which are present extensively in pharmaceuticals. Like pharmaceuticals are generally the combination of various enormous organic compounds. Majority of these complex organic molecules have low biodegradability and they cannot easily degrade in soil. They cause toxic and harmful effects to the environment. As we all know, there are various types of chemical sensors used for the detection of pharmaceuticals, in which we are monitoring the concentration of diclofenac as an example. So let us study the use of these sensors in detection of diclofenac. Now let us know the use of electrochemical sensor in the detection of diclofenac, which is also termed as DCF. The detection involves the interface between analyte and the working electrode where the reactions occur. In the detection of diclofenac, along with the sensing electrode, counting electrode and the reference electrodes are also used. In the detection of diclofenac, along with the working electrode and the counter electrode, there is the reference electrode also being used. Carbon paste with MWCNT. MWCNT is the multi-walled carbon nanotubes and our reference electrode is Ag bar AgCl. So, in the working of electrochemical sensor in the detection of diclofenac, we have this type of electrode system where the 3 electrode system is being depicted in the diagram, where the working electrode is made up of carbon paste with MWCNT and the counter electrode is made up of multi-walled CNT, that is multi-walled carbon nanotubes, as well as the reference electrode consists of Ag bar AgCl. Now, when the sample containing diclofenac is put in the sensor, then oxidation of diclofenac occurs on the surface of carbon. So, carbon electrode which releases the electrons. When there is reaction or the interaction of the analyte and the carbon electrode takes place, there is liberation of electrons which form radical intermediate, and also they causes hydrolysis of this radical intermediate occurs. Then the products are formed. The products formed are 2,6-dichloroaniline and 2,2-dihydroxyphenyl acetic acid. I'll show you the reaction in the next step. Whereas the reaction of the electrode system causes the current to flow, we know when there is a reaction occurring between the analyte and the electrode, there is the formation of this product where we get the liberation of electrons or the product and byproduct which is being formed, and then the intensity of current. So the intensity of current which is liberated or which is measured during the reaction gives us the concentration of diclofenac present in that particular analyte. Let us now know the reactions which are responsible for the liberation of electrons and the product formation. So these are the reactions which occur when diclofenac reacts with the electrochemical sensor. When a sensor is put into the analyte containing diclofenac, the diclofenac undergoes reaction forming a radical intermediate. This is the radical intermediate so formed. This undergoes hydrolysis to form 2,2-dihydroaniline and 2,2-dihydroxophenyl acetic acid. The formation of these two products, the formation of aniline 2,2-dichloroaniline as well as 2,2-hydroxyphenyl acetic acid, indicates the presence of diclofenac in the given analyte. The amount of product generated is responsible for the amount of change in potential or the amount of current produced. The amount of current produced is in turn responsible for the concentration of pharmaceutical, that is the concentration of DCF present in the given analyte. So this is the reaction of diclofenac which is being formed into 2,2-dichloroaniline as well as 2,2-hydroxyphenyl acetic acid. This is about detection of pharmaceutical diclofenac in using electrochemical sensor, using a 3-electrode system where we use working, counter as well as a reference electrode.